director of the Pharmacogenomics knowledge base, PharmJKB, at Stanford University. She leads the PharmJKB team and is responsible for the development of new content, projects, and futures. For the last 20 years, she has led pharmacogenomics research and its application to personalized medicine and personal genomics. She has leadership roles in multiple NIH-funded pharmacogenomic projects, and in addition from PharmJKB, including the Clinical Pharmacogenetics Implementation Consortium and the Pharmacogenomics Clinical Annotation Tool, and has served on multiple national and international pharmacogenomic working groups and steering committees. So her research interests include the translation of human genome sequencing data to clinical implementation using curated pharmacogenomics knowledge. So, Michelle, thank you.
Thank you. Thank you so much for the introduction, and thank you for the opportunity to speak with you all today. I really appreciate it. And I'm well aware that I'm standing between you and dinner for most people, so I'll try to move it along. Oh, just a disclosure really fast. I don't really have anything to disclose other than my public funding. So we had an excellent overview of pharmacogenomics from the previous speaker. Munir did a great job explaining it. So just going to start with talking about how the amount of pharmacogenomic information that we've had over the past 20 years has really gone up. And this is almost very similar diagram to what Munir showed, where I just started in the year 2000. There were only about 387 publications for pharmacogenomics that were found in PubMed for that year, versus in 2021, in that year alone, we've got over 2,600 publications. So we are accumulating knowledge all the time. There's over 35,000 publications in total in PubMed right now about pharmacogenomics and genetics. So it's great to have all this information. More and more information means that we are more informed, but just the information alone can be tricky. What do we do with that? So this raises some issues about how do we organize all this information and how can we standardize it across all these different publications? People use different terminologies, are measuring different things, and it's not standardized currently. It also leads to questions about how can we use this information for clinical actionability? So we really need centralized resources to help us be able to organize this information, standardize it, so that we can easily search across it. So I'm going to talk to you today about a few resources that are available that help centralize this information. But first I'm going to talk about a few sources of pharmacogenomic knowledge that we can accumulate together in order to get a better picture of the field. So one is the peer-reviewed published studies. I already showed you what's in PubMed right now, thousands and thousands of studies. We also know that there's some information in regulatory agency-approved drug labels. So you heard a little bit about that from Munir as well, that some labels from the EMA, for example, from FDA, do have information on them about testing for particular genetic variants or having a metabolizer status and how you can choose a drug or change the dosage accordingly. But that's a very few drug labels that have that information on them currently, although it is getting more and more all the time. We also have published guidelines. So there are different groups that write actually clinical guidelines for clinicians to how to go from the genotype to dosing the patient. Most of those guidelines are derived from the peer-reviewed published literature, though. So they're kind of a derivative of that data source. We also know that there's a lot of unpublished data out there, right? So there's clinical trials and other experiments that may be proprietary from pharmaceutical companies. That information is more difficult to use and to standardize, and also to vet. As a public resource, it's hard to deal with that information. But we know that a lot of that information is used in those regulatory agency-approved drug labels as well. A lot of that is submitted to these agencies when the drugs are going for approval. So one of the sources, centralized sources for dealing with organizing this type of information that I just discussed is PharmGKB, which is the Pharmacogenomics knowledge base. This is a website with a database backend. It's publicly available. It's probably the biggest resource that's publicly available in the world right now. And we started this around the year 2000. It's based at Stanford University. But our mission is basically to collect, encode, and disseminate pharmacogenomic knowledge for uses ranging from research and discovery all the way through clinical actionability and implementation. So what do we do at PharmGKB? We take the information that is out there that I kind of already went over, guidelines, drug labels, what's published in the literature, and we think of this as our knowledge information stream. This is all publicly available right now for everyone, but at PharmGKB, we have a team of scientists that extract information from these sources, and using expert manual curation, we take the relevant parts of like what drug, what genotype, what gene, et cetera, what variants. We standardize the terminologies, and we aggregate this information together. And we're always working towards clinical implementation and adoption. And in the end, we present some resources that I'm going to show you in a minute that actually go back out into this knowledge stream, and we hope enrich it and make it a better source of information for people who are trying to implement pharmacogenomics in their clinics. So one source of information at PharmGKB are these guidelines that I mentioned earlier. And the two groups that probably publish the most information about clinical implementation of pharmacogenomics would be CPIC and the Royal Dutch Pharmacists Association's Dutch Pharmacogenetics Working Group out of Netherlands. So on PharmGKB, this is just an example webpage. I don't expect you to read it. It's just an example. But so for example, the Dutch working group has some guidelines about the use of amitriptyline and how CYP2D6 variants can affect affect dosage of this particular drug. And the Dutch working group puts out a PDF that is text-based that people can read and get this information themselves. But what we tried to do at PharmGKB is make this a little bit simpler presentation through tables, and by using all the information available from the Dutch group itself. We have on the webpage a way for people to input what the particular variants are of a specific patient or an example, and pull up what the recommendation is for that specific genotype. So just a little bit of an aid for people to be able to take the information from the Dutch group and get right to the source without having to understand the mapping from the actual genotype in the gene to the metabolizer phenotype, such as poor metabolizer, et cetera, to the actual recommendation. So we have on our website just the broken down information from multiple groups, including CPIC, the Dutch group, and there are a few other groups that have some one-off recommendations as well. And you can compare across these. Due to resource issues and so forth, not all the same drugs have been have guidelines written about them from both organizations. And so sometimes one organization may have something about a drug that the other one doesn't. Most of the time, the recommendations agree, or at least are very similar, but sometimes there's a little bit of differences. And you can compare those at PharmGKB to see what the differences or similarities are. And we also highlight where guidelines tell you give you testing guidance, what to test and when to test. So another source of information that we organize would be drug labels. We annotate drug labels from FDA, but also from EMA, Canada, and we've had a couple of collaborations with groups in both Japan and Switzerland. So we have some labels that have been annotated from those groups as well, and we're always keen to collaborate with others. So if anybody is interested in working with us on that, please let me know. But our biggest group of drug labels would be from FDA and then followed with EMA. So We saw an example of a drug label earlier from Munir, and sometimes on drug labels they do highlight that there is information, but much of the time the language on drug labels can be not precise or somewhat vague. And so we had feedback from many of our users that what they really wanted to know what these drug labels is, just tell me which labels say I need to test, which ones recommend testing, and which ones have any kind of information that I can use. So we came up with this a labeling system where we can tag these labels that we curate with those categories. And also we get very specific if there is prescribing information based on pharmacogenetics on a drug label, such as changing the dose or changing the drug, we highlight that as well. So this is just an example screenshot, I don't expect you to read it, of annotation of the EMA label for abacavir and HLA-B. You heard a little bit about this earlier. So yes, this is a very kind of famous example of pharmacogenomics, very well understood. And so yes, testing is required according to the label. The label gives you specifics about what variant it is specifically in HLA-B that you should be interested in, and gives you advice about what to do. So this is an example of a nicely laid out drug label, and we were able to capture this information very easily, but not all drug labels are quite so simple. So again, we have a page at PharmGKB. Please go ahead and check it out for yourself at some point if you are able and inclined. We have drug labels from FDA, EMA, and a few other groups as well. And you can use this table to compare across, and you can drill down to see the exact annotation like I just showed you for the EMA one and abacavir. But what's interesting is that some regulatory agencies have different information on their labels depending on either they may be lacking information altogether about pharmacogenomics on that label, whereas other countries or groups have a very high level of actionability for pharmacogenomics. So it's kind of interesting to look at this webpage to see overall, again, where the similarities and differences are, and it just has to do with what is submitted to these regulatory agencies and what is deemed actionable at the time. So we also heard from Munir a little bit about randomized control trials. Again, yes, this is considered the gold standard, and a lot of clinicians prefer to have information that comes from RCTs, and this is where they would like to see any kind of proof of clinical actionability. But as was pointed out, it's really not feasible. A lot of times we have small populations in these studies. Again, Munir mentioned that. We don't have huge cohorts like in diabetes, et cetera. So the small populations do often make statistical significance difficult. But we do know that statistical significance is not always just the same as clinical significance anyway. And there is a lack of standardization, again, across all of these studies and publications that come out. But we can use replication of data from multiple different sources to help us address some of these issues, right? So we can take an article like this. This is from New England Journal of Medicine, and is talking about a particular association between a variant in SLC01B1 and a reaction to simvastatin. And if we take this and manually curate it at PharmGKB, again, we're standardizing across all the publications, the gene names, the gene variants. There are many different names for genetic variants, as I'm sure you're all aware, right? People can use RSIDs, they can use cDNA changes, protein, amino acid change, et cetera. So we can standardize so that we can see across all publications which ones are talking about the same variant. We can standardize the drug terms, et cetera, and we take a bunch of information about statistics and what type of study and the study size. We can collect that information from each article. And over time, there are more and more articles, right, about a particular association that can be published. Many times these articles replicate the original findings, but sometimes they do not. So sometimes an association is published and the next paper that comes out might refute actually that association. But if you look at a large enough cohort of papers, and like I said, you're able to compare across because of the standardization, we can start seeing the bigger picture and write summaries about the association based on the corpus of evidence. And then we can also take the information from those drug labels and the guidelines that I referenced earlier that are published and annotated at PharmGKB as well, and we can get an even bigger and better picture of what's going on with a particular association. Once we have all this evidence together, we can not only write a summary about what's going on, but we can assign a level of evidence about how confident we are in the strength of this particular association. So this is just a screenshot, again, for a particular association, the same variant with simvastatin. And down here, we can see that there are 2 dosing guideline annotations available on PharmGKB, one from CPIC and one was from the Dutch group. And in this case, we only have 9 publications, but there can actually be many, many more publications depending on the phenotype that's studied. So based on that grouping of information, we can come up with different levels of evidence. You can read more about the system that we have come up with for standardizing when we're trying to collect evidence together or visit the webpage. I can't go into a lot of detail here because it would take a while to explain. But basically, we score the information that we take from every single publication. We take that information and we add it together across all the different publications, and taking into account if any regulatory labels are available and the information on those labels, and also guidelines if they're available. And we come up with a total score for that particular summary annotation. Then based on that score for the summary annotation, we can figure out which cutoff for level that it meets and assign accordingly. So for all of these summaries, you can go, if you're on the PharmGKB page, you can see, get a feeling for how much support there is for a particular association based on the little labels on the left-hand side that are color-coded and also have the level written in them. In most cases, the level 1 are instances where we know of a guideline or a label that really confirms basically actionability or clinical implementation can be instituted for a particular association. And level 2 would be those that are very close, but maybe don't have a guideline or a label yet, but information on a label, but there's a lot of information published and the association looks very strong. Some people, on the other hand, are very interested in low-level associations if they're not looking to implement clinically, but they're looking at from a research perspective. Okay, so I'm going to switch gears for just a couple minutes to tell you a little bit more about CPIC. Again, this is one of the groups that writes guidelines for how to implement pharmacogenomics in the clinic. The Dutch group also writes guidelines as well. I happen to be involved in CPIC, so I'm going to give you a little bit of the background of that particular group. So the goal of CPIC is not to tell people whether or not to test or even what to test per se, but if you are a clinician and you have genetic results in hand, we want you to be able to understand and quickly be able to know what you can do with that information. Preemptive genotyping is becoming more widespread. There are direct-to-consumer genotyping companies out there, and some patients are going to their doctors, at least in the States, with information about their genetic variants, and they want their doctors to be able to understand that information and act on it. But most clinicians, as we heard, they don't have the time per patient to sit and really research for themselves what a particular genetic variation might mean or might imply for a particular drug prescription. So they need a facile way to access information that can tell them up front what it is that they should do. So these guidelines are written by expert groups put together. These groups include clinicians, gene experts, pharmacists, and in some cases, just research scientists as well. And again, we start with a PubMed literature review. So these guidelines are based on PubMed literature reviews. But the information is collated together and graded for every single outcome by the clinical authors, the authors of the guideline. And so a consensus is reached from that particular group, and then a statement about what you could do with a particular genetic test result. So, as part of this process, we have to understand for every genetic allele what the functional implication is. Somebody had mentioned that before. So yes, we do have to care about what the function is for all of these variants, and so that's part of the process for creating CPIC guidelines, is to look at basically do a deep dive literature review of every allele for the particular gene in that guideline and see if we can come to agreement about what the function of that allele is. Then once we have a function defined for an allele, we can put the two alleles together and map to a phenotype, such as a poor metabolizer or an ultra-rapid metabolizer that you heard about in the previous talk. Once we have the metabolizer status for a given genotype, for a given patient, then we can come up with what the therapeutic recommendation would be for that particular phenotype. So CPIC also makes available clinical decision support flowcharts and wording for CDS alerts to help aid with clinical implementation for groups that don't have the resources to do that as well. So this is just example charts and CDS language that people could use if they want to implement in their institution. So there's a bunch of CPIC guidelines right now. Last count was 26, but that might have changed really recently because we're always publishing. But please go and check out the website for yourself. We also have a database and API for people that are interested to access the information computationally and import it into their own databases. So another challenge with implementing pharmacogenomics would be our nomenclature. You heard a little bit about the star allele nomenclature already. I get people ask me all the time who aren't in the pharmacogenetics field, but more clinical genomics, what's the deal with the star alleles? Why do you guys have them and what do they mean? So just really briefly, right, we have the symbol, the gene symbol, and that star with the number after it is just an allele designation. And so for example, this CYP2D6 star 8, this is the HGBS representation for it. So there are, three different variants across the gene that define this allele. So in pharmacogenomics, with most of the cytochrome P450s, we're interested in what the combination of variants are across the entire allele. It's not necessarily one particular variant or another, but what's the combination of variants across the entire gene. It's the haplotype that matters. And so to define the haplotypes, the star allele shorthand was born. These variants, these are SNPs, so that's quite simple, but in many cases the variation across the gene can include repeats or indels. There can be structural variants and copy number variation as well. And the variation occurs not just in the exons, but also introns or upstream or downstream of the gene sometimes. And they all have functional effect and are very important to capture and understand what the variation is across. And so this shorthand helps the community understand what the variation is. I've been asked many times, will star alleles can they just go away? From people who are, clinicians but deal with Mendelian disease many times or other kinds of genetics. And unfortunately, I don't think that's going to happen. So it's used throughout the literature. Lab tests and assays, test reports all refer to the star allele nomenclature right now. And it's used by many of the prescribing guidelines. And this started in the 1990s. And was organized to a great extent in the early 2000s by the Human Cytochrome P450 allele nomenclature database in Sweden. And I think they were the first group to really start tracking these alleles and defining them as what variation is in each allele. And they're the ones that started naming them with the star alleles. And that has transitioned in 2017 to a group called PharmVar. The PharmVar group has taken over the reins there. And so PharmVar, please check out this group as well. So when we're talking about standardization, PharmVar is critical to standardization for these pharmacogenetic alleles. And it is the central repository. People who find new allelic variation can submit that variation to the PharmVar group. And there's a very rigorous process, rules that must be followed for a new star allele to be assigned. But if that's the case, if the submission reaches the level of a new star allele, it will be assigned. So just to show you a little bit about this is not the most complicated example by any means, but just to show you a little bit what I'm talking about. So this is, again, a CYP2D6 illustration. We're looking at the star alleles, star 10, 36, 37, et cetera. And then this is a list of variants down the side. And so what we see here is the 100C to T variant is a part of all of these different alleles. So is this variation down here. So if you, for example, we're just going to test this 100C to T, you might not really know which star allele you're talking about. For simplicity's sake, many times people just call it a star 10 because, okay, that's the defining variant in that particular star allele. But you can see that there's many, these other star alleles also contain that variant and then other variants as well. So it can get much more complicated than this as well. And some genes such as CYP2D6 have structural variants, and it gets crazy. But these star alleles really are important for understanding what the variation is, and that all has functional implications. So the functionality of these different star alleles can and do differ. So that leads to one of the other challenges of pharmacogenomics and implementation, is that the type of the test, what is tested, can affect your results, right? So there have been studies done where they've sent the same samples out to different labs and gotten different results back. And that a lot of times has to do with whether you're talking about a panel or an exome or whole genome sequencing, as someone had brought up earlier. And it's true that if you have a panel and not everything is on that panel that you can see in whole genome sequencing, you could potentially miss genetic variation that exists in that particular patient. And that's why we have to be really careful with this. Often in the field, if you don't see any variation, it is defaulted to what's called a star 1 allele, which implies that you don't have any genetic variation or wild type. But of course, depending on what's tested, that may or may not be true. So just something to keep in mind when people are trying to implement pharmacogenomics, that transparency about the tests and the test result can be very important. There can be situations where a patient is tested for pharmacogenomic alleles and none are found, and it goes into their the EMR or their record that they have no genetic variation in a particular gene, but we know that that may not always be 100% true, right? For example, there are some known existing genetic variations where we know of the genetic change, but we don't know what the function is. We don't know what to do with that change, but over time, research is going to eventually elucidate that for us. Also, there's new variation out there that may not be covered. So the current star allele nomenclature can't cover everything that's not known yet. So as new discoveries happen, they're submitted, and we have new star alleles that may not have been tested for previously. So at the very least, and what the results are can be very key to both clinicians and patients, especially going forward in the future. Another project that I'm involved in is called the Pharmacogenomics Clinical Annotation Tool. This is a software where the goal is to take the output from a genetic test report, the output from a genetic test such as a panel test or whole genome or exome sequencing, come up with the star allele designations for those genes, which can be very complicated, as was mentioned earlier. So this software aims to be able to take whole genome sequencing and determine that without using those tag alleles that are used on the SNP panel, and then connect the resulting genotypes with clinical guidance. And we've started with the clinical guidance from CPIC, but we'll be expanding that to other groups as well, such as the Dutch guidelines and also what's available on regulatory agency drug labels. And that's the goal. So basically the way this tool works is it takes a VCF file, which is just a way that genetic variation can be output from testing, and then we take those allele definitions, like from PharmVar, that has the definitions of what variants are in each star allele, and the information from the guidelines that can be accessed via the API in the database. And then we also have some messaging from the PharmCat tool as well, with caveats and disclaimers, et cetera. And we take all of that together. So what's the user supplies is the actual genetic variation that are detected. And we come up with a genotype summary, which tells you what the star alleles, what the diplotypes are or genotypes for that particular patient based on that VCF file, and give you a summary of the output, then the recommendations by drug. So you may be a CYP2D6 star 4 star 8, for example. What does that mean? We have recommendations for each drug that we know that can be affected, or have guidelines showing that the drug response can be affected by CYP2D6, and then we would tell you exactly what the recommendations there are for that particular genotype. And then there's a section of the report that is very key, but not necessarily, first and foremost in any clinician's mind or patient for that matter. But very importantly, we highlight what variation was given to us in the VCF file versus what is known to date. So we can easily see in this section of the report, if not every known genetic variant that could be part of a star allele, et cetera, was tested. So in this way, we can see with an assigned genotype at the end, if information was essentially missed, if there was known star alleles that were not tested for. In this case, you would know then if, say, a patient is a star 1 as the result, whether or not there are potentially variation that patient is carrying in that gene that was not covered by that test. So it's great to have this as a report. You get a report out of PharmCat. And that's a report that a patient could have and take to their doctor, or a clinician could look at. But many times, as we know, clinicians don't have the time to sit and read a report either. So having this information go automatically into an EMR or EHR system is extremely important, and that is a big challenge since they are not standardized across all hospitals or hospital systems. And so that's another area that we're working on for the future. One of the final challenges that I'm going to talk about today would be allele frequency, and this again was mentioned a little bit earlier. But the issue is that in many cases, a Mendelian disease, a low frequency is a sign of maybe pathogenicity, right? So frequency can be used as a way to figure out if something might be very important in a particular gene. But unfortunately, in pharmacogenomics, some of these variations can be quite common, up to 30% or even more in certain populations. So you cannot use frequency the same way in pharmacogenomics as a sign for what could be affect function. And yet, knowing the frequency of the alleles is very helpful to know, for example, if you can only test a handful of alleles, which ones should you be testing? Unfortunately, we don't know the frequency for many of the defined alleles. They may have been discovered in a particular patient, submitted to PharmVar, for example, gotten a star allele. Now we know we can test for them. But if people haven't tested for them previously, we don't really know what the frequency in any given population is. So many times we think in pharmacogenetics in the field, we think certain star alleles are rare because it's not published much in the literature. We can't find statistics about it, but that's not necessarily the case. So we do have to be careful there. Also, many populations around the globe are understudied. So again, we want to make pharmacogenomics available for all diverse populations, but we haven't done a lot of studies yet in many of the populations around the globe. A lot of them have been focused on European populations or perhaps Asian populations as well. So research is needed there to understand what the frequency is for these variants, but we always must remember that when we're trying to implement this, the population frequency is never a proxy for what a patient actually has. So testing the individual patient is really important. So the last challenge I'm going to talk about is just the separation of pharmacogenomics from the rest of clinical genomics. I gave you a little bit of an overview of all these four resources: PharmGKB, PharmCat, PharmVar, and CPIC. These resources talk to each other all the time. They trade information back and forth. In the US, we have very big projects for clinical genomics. ClinVar and ClinGen are huge resources that are funded through the NIH. But they're kind of isolated. Isolated from pharmacogenomics. I should say pharmacogenomics is isolated from them because they are very large and capture most of the genetic clinical genomics that are documented today. But pharmacogenomics kind of exists as a silo. We are trying to address that by depositing information from both CPIC and PharmGKB into both ClinVar and ClinGen. There are some issues with trying to take pharmacogenomic information and wedge it into a clinical genome database. And so it's a little bit tricky at times, and the deposition of this information is slow. But in addition to depositing it, we are looking forward to being able to exchange information with ClinVar and ClinGen as well. So what we really want to do is not have pharmacogenomics be isolated and siloed from clinical genomics overall and genomic medicine. And our vision, right, would be full integration of pharmacogenomics with genomic medicine. And this is just a diagram to try to illustrate that all those pharmacogenomic resources and more could, eventually be combined together in some way to have a much easier interface, maybe one place that people can go, and that group could interact very closely with both ClinVar and ClinGen. And efforts are underway. We have a panel that we're starting within the ClinGen group to address pharmacogenomics and how to better crosstalk between pharmacogenomics and genomic medicine in general. So in many cases, we don't know the implications of pharmacogenomic discoveries for clinical implementation, but we definitely know that the clinical utility is there for a couple dozen examples at least. And then we know that integrating this together with the rest of genomic medicine is going to push implementation forward. Having to try to address this separately is slow going. As Munir described, it's not being implemented across as many places as we'd like pharmacogenomics to be yet. It does differ from genomics disease models. The haplotypes and the star alleles and the fact that you have to be aware of the diplotypes and map that to metabolizer phenotypes in many cases is very different, and we are aware of that. So we do still need some specialized resources, but integrating would be the way to go in the future. And as of right now, we have a few centralized knowledge sources that people can use in the meantime. And with that, I'd like to just thank all of my colleagues. There was many different projects that I was talking about today, and it takes many people in many different institutions to help make all these projects a reality. And so I'd like to thank them, and thank you for your attention and your time.
We have questions. I will start from the chat. One question regards SCLO1B1 and statins, and basically what they ask is, are there studies regarding drug adherence and pharmacogenetic testing or genotyping? I mean, does it influence adherence of the patients?
Adherence, yes. I'm not aware of specific studies about adherence to drugs, but we know colloquially, just anecdotally, that this is true, and that patients who have adverse effects that are not as severe as like what we saw in the previous talk, but just myopathy, maybe pain or some other kind, nausea, etc., can lead patients to stop taking their medication. And so, yes, it has been shown that because of the myopathy, that's associated with a particular genetic variation. There have been cases, like I said, anecdotally, where patients just stop taking their medicine because of it.
Okay. Yeah.
Hi, Michelle, over here. Thank you. Great presentation, fabulous resources, and I think incredibly useful for the community. Can I just ask, are there plans or thoughts about translating those resources into different languages to make them more accessible?
That is an excellent question. We would love to do that. Unfortunately, resource-wise, being government funded, we don't have the resources to pay for that. But if there are people that would like to collaborate with us, we could see what we could do. Yes, we get that question a lot. You know, can this be in multiple languages? Happy to do it. Just a resource issue right now.
Okay, another question from the chat is, despite the fact that pharmacogenomics data is growing rapidly and the importance of testing is clear, the integration in the clinics is very slowly. So maybe you could comment on the barriers or the reasons for that.
Right. Yes, I think a lot of the reasons for the slow uptake were mentioned previously. Just having the education, I think, of clinicians for them to be familiar with this type of data, familiar with how to use it. Also, there are issues putting this kind of information in medical records, so getting this into the EHR system, I think, would be key. That's one barrier we know to uptake. And then just also, testing and reimbursement. What are you supposed to test for? When should you test? Who's going to pay for it? These kinds of issues definitely affect implementation as well.
Okay, one last question. Yeah.
Thank you as well for a great presentation. We in our company started testing the Pharmcat, and I'd like to ask how many drugs are in this moment supported, because we have only a limited number, and also if it can be adjusted by the users,
yes,
the parameters, and what are you going to expand it?
Yeah, absolutely. So yes, so if you check out pharmcat.org. So we've updated a lot of the documentation, maybe since you have checked it out. All of the drugs that are covered by CPIC guidelines are currently covered by Pharmcat and are in the report. All the genes, and yes, you can alter. Well, first of all, it's freely accessible. So if you want to download the tool and branch off, you can change the code however you like. But it is also customizable in terms of if you have genes that you want to include and you have the definition files for those, you can can add those as you want, and same with the recommendations as well. And we are going to be expanding those within the next year is the goal, to include drugs and recommendations from the Dutch Pharmacogenetic Working Group, and also what information we can find on the FDA labels. We'll probably start with FDA, but then ultimately can expand from there as well.
Okay, those are bioinformatics that are working in our company on it, so I'm not completely familiar, but is it possible to speak to your ITs?
Yeah, sure. You know, send, I didn't have the email up here, but if you send email to just feedback@pharmgkb.org, we will make sure it gets to PharmCat. I think there's also pharmcat@pharmgkb.org, but I can't swear to that. So if you send it to feedback@pharmgkb.org and you say this is a question about PharmCat, we can deal with that. We're a very small team, and we are the same people working on many projects at once. But but yes, definitely.
Thank you.
Thanks.
Okay, thank you. So I want just to remind you that if you continue,